Hello students, today we will discuss about the chapter of biodiversity and conservation. This is chapter 15 of the NCERT and these are the notes which is prepared by me, Dr. Ramji Chopra. These notes are according to NCERT, so please follow them in order to succeed in your exams. So what is biodiversity? Definition. Variety and variability of life on the earth is biodiversity. The term was popularized and coined by social biologist Edward Wilson. Now there are different levels of biodiversity. Number 1, genetic diversity. Number 2, species diversity. Number 3, ecological diversity. Genetic diversity, it refers to diversity of gene within a species. Example, there are more than 50,000 genetic different strains of rice in India, number 1. Number 2, there are 1,000 varieties of mango in India. And number 3, it is also represented by Rovalfia in the terms of potency and concentration of active chemical reservoir produced by the plant. Number 2 is species diversity. It refers to number of different species within a given region. Example, Western Ghats have greater amphibian species diversity than Eastern Ghats. So example of species diversity is Western Ghats have greater amphibian species than Eastern Ghats. Ecological diversity. Now, ecological diversity means variation of habitat, community types and abiotic environment. For example, India with its deserts, rainforests, mangroves, coral reefs, it has a greater ecosystem diversity than a Scandinavian country like Norway. Now, on the basis of this text from NCERT, we have the following questions. Who coined the term biodiversity? So its answer is Edward Wilson. Next, we name the type of biodiversity represented by the following. 50,000 different strains of rice in India. So its answer is genetic diversity. And estuaries and alpine meadow in India, its example is ecological diversity. Now, name any two levels of biodiversity. Here you can describe both species diversity, ecological diversity and genetic diversity. Next is, why is genetic variation important in plant welfare? Now, genetic variation is important in plant welfare because it consists of active chemical reservoir, which is shown in the terms of potency and concentration by the plant at genetic variation. This chemical reservoir is shown in this chemical reservoir. Potency and concentration is different in different strains of Rovalfia. So it is an example of genetic diversity. Now come on to the next page. How many species are there on the earth and how many in India? So there are 1.5 million of plant and animal species in India. According to Robert May, the biodiversity is about 7 millions. Total species, 70% are animals, 22% in plants. Of animals, 70% are insects. That is, out of every 10 animals, 7 are insects. Now these are the diagrams which are very important. You need to memorize this. Now next question which comes is, why are prokaryotes not given any figure for their diversity by the ecologists? Because the conventional taxonomic methods are not suitable for identifying these microbes. Many of them cannot be cultured under laboratory and molecular biochemical techniques would put their diversity into millions. So we do not have any taxonomic method for identifying these microbes, nor they can be cultured in the laboratory, nor molecular and biological technique put the diversity into million. If we will study biological and molecular technique, it would put their diversity into million. Now, India biodiversity. India has 2.4% of the world land and 8.1% of the total species diversity. According to May estimate, 78% biodiversity is still to be discovered in India. Now, on the basis of this, here are the questions. So, identify A and B in this diagram. So if we look at this diagram, this is of the vertebrate taxa. So this is the diagram of vertebrate taxa. So A is mammals and B is amphibians. give any two reasons as to why prokaryotes are not given any figure for their diversity by the ecologists. So prokaryotes are not given any figure because these are the reasons. The conventional taxonomic method are not suitable for identifying these microbes, and many of them cannot be cultured in the laboratory. So if you like this video, in the next video of biodiversity will be continued in the next video. Subscribe now to my YouTube channel. Thank you.